Hi friends, so now I'm back with my next video, and in this video we are going to talk about nucleic acid. So we are going to talk about what are different types of nucleic acid, what is their structure, the bonding between them, and what is their role in the replication of the DNA replication and then in the transcription and translation. So please make sure to like, share and subscribe my channel. So, what is a nucleic acid? It's a molecule that stores information for the growth, reproduction to perform different function. So basically, these are the hereditary unit that get transferred from the person to person, right? From generations, and it stores the information like what are the proteins that it codes for, and different type of transcription factor as well. And basically, there are two types of nucleic acid, the deoxyribonucleic acid, the DNA, and the ribonucleic acid. So the difference between the both of them is the presence of the hydroxyl group in the ribonucleic acid and the absence from the deoxyribonucleic acid. And they are the polymers, like both of them consist of the long chains of the monomers, which are called nucleotides. So what is a nucleotide? So here you can see we have a nitrogenous base, which we will talk about is what is a nitrogenous base. And then it consists of a sugar molecule, pentose sugar, which is different for the DNA and RNA, because this sugar in case of the DNA will be the sugar without hydroxyl group, and which is deoxyribose, and in case of the RNA it should be the ribose, and then a phosphate group which is linked to this sugar using the ester bond. So this whole complex makes a nucleotide. So the nucleic acid, either DNA or RNA, consists of long chains of these nucleotides. So what is a nitrogen base or the nitrogenous base? So it's the base in nucleotide. It consists of two types, purines and pyrimidines. So there are two types in the purines, adenine and guanine, and in the pyrimidines, cytosine, thymine and uracil. So here you can see the purines, which consist of the double rings, one 6 carbon rings and the other 5 carbon rings. So they are called the purines, adenine and guanine, and then the pyrimidines consist of only one ring, cytosine, thymine and uracil. So in case of the DNA, it consists of the nucleotides like guanine, adenine, cytosine and thymine. But in case of RNA, thymine is not present. So instead of thymine, we have the uracil. Okay, so we in both of them we have four bases like the guanine, adenine, cytosine, and thymine in case of the DNA and uracil in case of the RNA. And then what is a pentose sugar? So as I have already talked about, there are basically two types of pentose sugar in nucleic acid. The RNA contain the ribose sugar and the DNA contain the deoxyribose sugar. So here you can see this ring, right, with the first prime carbon is this one, and then we have like the five carbon sugar chain, and then here you can see the difference between ribose and deoxyribose is the presence of this hydroxyl group. So in case of ribose we have this hydroxyl group, but in this case of the deoxyribose, the sugar which is present in DNA, we don't have the second carbon hydroxyl group. That's why it's called the 2-deoxyribose, and this is what makes a difference between the DNA and RNA. So now we have talked about nucleotides. So now we need to know what is a nucleoside as well. So nucleoside, as we have talked about, it consists of a nitrogenous base, which can be, as we have talked about, any of the four bases, adenine, guanine, cytosine, thymine or uracil, and then a pentose sugar ring which is attached to this nitrogenous base. So this is called the nucleoside. And when we add a phosphate to this group, it makes a nucleotide. So the difference between nucleoside and the nucleotide is the presence or the absence of the phosphate group. So now what is the structure of the nucleic acid? So as I have talked about, like the primary structure is the like the linear structure that we are talking about is the nucleotide sequence, a long chain of monomers of the nucleotides, and the different nucleotides like first nucleotide, second nucleotide, they are joined together by the phosphodiester bond. So here you can see we have one nucleotide, and then this is the another nucleotide, and when they need to combine, so there is formation of the phosphodiester bond. How does this happen is the phosphate group of one of the 5 carbon sugar, it forms an ester bond with the 3 carbon hydroxyl group. So here you can see this bond with the phosphate and the hydroxyl of the sugar, right? 5 carbon and the 3 carbon of the sugar, and it forms an hydroxyl it forms a diester bond, and then more nucleotide can be joined using the 5 phosphate group here and the 3 hydroxyl group here. So then it forms a long chain of the nucleotides, which generally forms the primary structure of the nucleic acid. So as I have talked about, it always has a free 5 phosphate group and the free 3 hydroxyl group, which can which can form the polymer by adding different monomers using the 5 phosphate group and the 3 hydroxyl group, and the sequence is read always from the 5 end. So here you can so we have the direction like from 5 to 3, right? 5 phosphate group and the 3 prime hydroxyl group. So the sequence is always read from the 5 end using the letters of the bases, not from the another direction from like 3 to 5. It's always read from 5 to 3. So we have talked about the primary structure, which is a linear structure of the nucleic acid, but the nucleic acid, like the DNA, it consists in the DNA double helix structure. So there are 2 different strands of the DNA that come together, form the complementary bonding between them to form the double helix. So as I already talked about, in DNA, the 2 strands of the nucleotide that comes together wind together to form a double helix, and the strands run in the opposite direction. What does that mean is, suppose you have one strand with the direction from 5 to 3, the another strand should be from 3 to 5, then only they can combine, because 5 and 5 cannot combine with each other. It needs to be 5, and then the another strand needs to be 3 to 5 direction, and the bases are arranged in a step like pair, like we know that, like we have already discussed, and the base pairs are held together by hydrogen bonding. So like there are 2 strands. Suppose in one of the strand we have the adenine, in the another strand we should have thymine, and then these two forms a hydrogen bonding between them, right? And then these 2 strands, they are always arranged by the hydrogen bonding. So the complementary base pairs are AT and GC, which means adenine and thymine, guanine and cytosine. So adenine is a purine and thymine is a pyrimidine. So one purine makes the bonding with the one pyrimidine, and same with guanine and cytosine. So it is not possible that adenine is forming the bond between guanine, or thymine is bonding the bond between cytosine, or vice versa, or with others. Adenine will always form the hydrogen bond with the thymine. And there are 2 hydrogen bonds which are formed between the adenine and thymine, and 3 hydrogen bonds which are formed between the guanine and cytosine. And as I had talked about, each pair consists of one purine and one pyrimidine. So they are the same width, like one purine and one pyrimidine. So they keep the 2 strands at the same width, so that there is no the one of the area which is of bigger width, the other area of smaller width. They keep the same width, and they are at equal distance from each other. So now we have talked about the deoxyribonucleic acid, the DNA, which is consist of different 2 strands, different bases, how these bases are combined. So the important thing about RNA is it consists of a single strand. Instead of DNA, which consists of two strands, the double helix, the RNA is a single helix. And RNA is much more abundant than DNA, because from one DNA you can form different kinds of RNA. So it's much more abundant. So there are some differences between the RNA and DNA. The first difference we have already talked about is the sugar. One contains the ribose and the other one contains the deoxyribose. And in RNA, uracil replaces the thymine. So U pairs with A instead of T pairing with A. And RNA is single stranded while DNA is double stranded. And there are basically three types of RNA: ribosomal RNA, messenger RNA, and transfer RNA. So I will talk about what are their functions. So ribosomal RNA, as the name suggests, ribosomes are basically the sites where the protein synthesis occurs. So where proteins are formed, and then different amino acids combine to form the protein, and then it passes through different secondary structure, tertiary structure. So this ribosome, it consists of many ribosomal RNA and protein. So they basically have two subunits, the large one and the small one. So the RNA, so the ribosomal RNA, it is involved in this translation, right? So it plays some role in the translation and in the ribosomes. And the messenger RNA, it carries the information, the genetic codes to the ribosome. So now first we have the DNA, right? From DNA it forms a RNA, or called the messenger RNA. And then this messenger RNA goes to this ribosome subunit or ribosomes to form the protein. So messenger RNA carries the DNA genetic information to the ribosome, where ribosomal RNA and proteins assist in the ribosomal activity to form the protein synthesis. And then during this protein synthesis, there is and the messenger RNA, as I have talked, they are complementary to the DNA, right? And then another important RNA is the transfer RNA, the tRNA. So what does it do is, so you have the ribosome where protein synthesis occurs, but you need the specific amino acid based on the information, right? So transfer RNA, it translates the genetic code from the messenger RNA, and it brings specific amino acids to the ribosome. So you see the information, like there are three bases, right, which forms a codon, and then the transfer RNA, it recognizes this basis and brings a specific amino acid based on that codon, right? So now the information about this is clear, and all the RNA, what does it do? So I hope it's clear, the basic knowledge about the nucleic acid. If you have any doubts, please let me know in the comment section. And in my next video, I will talk about in detail the replication of DNA and the transcription and translation. So please make sure to like, share, and subscribe my channel. Thank you.